Welcome children. Let us see lesson 4, Principle of Inheritance and Variation. What is inheritance? Inheritance means inborn. Inborn are the character received from the parents. And what is variation? Variation are the changes. These variations are brought through the gene as a character it received from the maternal mother and paternal father by the exchange of gene. So we can observe the progeny or offspring or young one with different character from the parent. Now let us see what is genetics. Genetics is a branch of biology that otherwise said to be living science deals with the study of heredity and variation. What is heredity? Heredity is the character which pass on from one generation to another successive generation through the gene. For example, you can see some of the behavior or character of the progeny such as singing and dancing received from their grandparents through the gene. What is variation? The variation is the changes by which the progeny differ from their parent. Progeny means young one. Now let us see what is allele. Allele is a pair of genes controlling the same character and located at the same locus in the homologous chromosome. What is locus? Locus is nothing but a specific place in the chromosome where the gene is located. We can't observe the gene in all the places. In particular places where the gene is located is said to be as the locus. In homologous chromosome, what is homologous chromosome? Homologous means identical, similar or sister chromatids. Now coming to the multiple alleles. I told in the alleles are the pair of genes controlling the same character and located at the same locus in homologous chromosome. But in multiple alleles are set of 3 or more genes controlling the same character located on the chromosome. Let us see some reference for the multiple alleles. They are human blood group, sex determination in man, insect and bird, sex-linked inherited triads, genetic disorder and extra chromosomal inheritance or the example or the reference for the multiple allele. For the betterment of the human being or human races, they achieved several methods like eugenics and euphenics. What is eugenic? Eugenic is a science of well-born, improving with the inborn qualities of races and obtaining the better heritage by judicious breeding. What is euthenic? Euthenics deals with the importance of already existing human being by subjecting them with better nutrition, education and medical care. What is euphenic? Euphenic deals with the control of several inherited human diseases. That is, example for that disease is inborn metabolic error disease, phenylketonuria. So, these are the methods adapted to achieve the betterment of human races. Now, it is about the 2 alternative alleles, dominant and recessive. The dominant allele is represented by capital T and the recessive allele it is represented by small t. Always the dominant allele is normal allele or wild allele. The recessive allele is said to be mutant allele. A gene can mutate several times producing several alternative forms. In the multiple allelism we have seen that when 3 or more alleles of a gene that control a particular triad occupying the same locus on the homologous chromosome of an organism is called as the multiple allele. If it is inherited, it is said to be as a multiple allelism. You can observe the picture showing homologous chromosome. The homologous chromosome, one is said to be paternal and another one is said to be as the maternal. That is, maternal means mother, paternal means father. So in this we can see one is representing the dominant allele capital T and another one is representing the small, that is dwarf recessive small t. So small t is said to be as a mutant allele. Human blood group is one of the examples for multiple allele. The human blood groups are distinguished according to the presence and absence of the antigen and the antibody in different person. So according to the presence of antigen and the antibody, the human blood groups are classified. Now let us see what is antigen. The antigen is otherwise said to be agglutination. That is nothing but the blood group gene which determine the synthesis of polysaccharide on surface of the erythrocyte or red blood corpuscle are known as the antigen or agglutinogen. Now coming to the antibody, it is a specific substance or the protein produced in the blood serum in response to the stimulation of the foreign antigen and react specifically to the particular antigen. The RBC showing antigen and antibody. What is agglutination? See, in the transfusion, if the blood of the donor and recipient are not compatible, agglutination and hemolysis of donor corpuscle will take place. What is agglutination? It is clustering of RBC. What is hemolysis? Death of RBC. So, it is very important that the reaction between antigen of the donor and the donor's RBC and the antibody of the recipient plasma, it is very important. So it is due to the agglutination will happen due to the surface antigen on the membrane of RBC and epithelial cell. So blood differ chemically from person to person. When two different incompatible blood are mixed, it leads to the death of RBC and clustering of RBC. That is said to be as the agglutination. According to the presence and absence of the antigen and the antibody, the blood group is distinguished into A type, B type, AB type and O type. So there are 4 types of blood groups. How the blood groups are grouped? According to the antigen and the antibody. So if a A type blood group, it is considered with the antigen A and antibody B. If it is a B type blood group, it has the antigen B and antibody A. If it is a AB blood group, A and B antigen are found to be present, antibody is absent. If it is O type blood group, it won't have any antigen, it have only the antibody and A and B. Now who discovered the blood group? The blood group is discovered by Karl Landsteiner and his student Van de Castel and Sterling. They both in 1902 they discovered the 4th blood group, that is AB. So now coming to the donor and the recipient. So what is donor and what is recipient? One who donate the blood is said to be as a donor and one who receive the blood is said to be recipient. Here it is about universal donor. Why it is said to be universal donor means the universal donor has the capacity to donate blood to all the A type, B type and AB type, but it won't receive blood from anybody because it lacks antigen and it has only the antibody A and B. And the universal recipient AB, it can receive the blood from all the groups such as A group, B group, AB group and O group. It won't donate blood to anybody. So it is said to be universal recipient because it has the antigen A and B, antibody A and B are absent. Scientist Bernstein in 1925 discovered that the inheritance of, that is inborn of, different blood group in human being is determined by a number of multi-allelic series. What is multiple allele? A set of or more than 3 genes located in the same locus determining the same character of homologous chromosome is called as multiple allele. So the multiple allele in a series is going to determine the blood group of the person. It will be differ from person to person. Coming to the chromosome, in human there is a constant 23 pairs of chromosome. In the 23 pairs of chromosome, autosome, which is determining the body character, is found to be 22 pairs. The body character, it may be height, it may be the hair or color, it will be determined by 22 pairs of chromosome. The remaining 1 pair of chromosome is said to be allosome. It is determining the sex. So allosome is otherwise said to be the sex chromosome. In the allosome, if it is female, it has XX chromosome, and if it is male, it is the XY chromosome. The Bernstein, he determined that 3 alleles which are found to be present in the chromosome number 9 is going to mention the blood group. So the blood group is determined in any person with the help of 3 autosomal allele that is located on the chromosome 9. Who discovered the blood? Landsteiner, he only discovered the blood. In order to honor the Landsteiner, they want to label the gene with the capital letter or otherwise with the capital I. I is nothing but the isoagglutination. What is isoagglutination? Isoagglutination is a process that occurs if any antigen is mixed with the corresponding antibody, that is called as isoagglutination. So according to the allele, the blood group is named with IA, IB, IO. The IA, it specifies the A antigen. IB specifies the B antigen. IO, it is not having any antigen. So according to the presence of antigen, the alleles are classified. Enzymes. What is enzymes? Enzymes are the catalysts for biochemical reaction in living cell. That is nothing but it helps to speed up the reaction. So in the blood also, each allele, that is IA and IB, it produce enzyme transferase. Now let us see the IA, it is producing the enzyme N-acetyl galactose transferase, which add N-acetyl galactosamine. IB, it is producing galactose enzyme which is added to galactose, that is H substance. H substance means histocompatible substance. IO, it won't produce any enzymes because it is said to be recessive null allele without any enzyme to add. So enzymes are very important for the life, biological life. Here I'll say one of the enzyme which is separated or isolated from the urine, that is called as ureaginase, that helps to dissolve the blood clot in the brain. So that much the enzymes are very important. Evident from the phenotypic combination by the alleles given by IA, IB and IO. IA and IB are found to be dominant over the IO. That means IO is recessive. IA is equal to IB, that is said to be codominant. What is codominant? Both the allele of the character are equally dominant. Both of them, they will express their character. That is said to be as the codominant. So the dominance hierarchy of the allele series can be symbolized as IA is equal to IB, that is lesser than IO. So IO is always recessive, it won't determine the character. Now let us see what is genotype. Genotype is the character expressed by the gene. Here for A group blood, we can see the alleles IA or IA-IO. For B group, we can observe IB-IB, IB-IO. For AB group, it is IA and IB. For O group, it is IO-IO. So these are the genotype which is expressing the gene for the blood group. Now coming to the antigen. What is antigen? Antigen is a foreign body or foreign protein. A type, it has the antigen which is representing in the human as a A-type blood group. It is observed in chimpanzee and in gibbon. So A-type antigen is found to be present in chimpanzee and in gibbon. B-type antigen, it is observed in New World monkey, Platyrrhina and lemurs. AB and AB antigen is observed in orangutan. In cat, there are 3 blood groups similar to the human have been distinguished. Now coming to the secretor. The secretor is nothing but the secretion which is obtained in the animal. Urine, tears, saliva, semen, gastric juice and milk of animal. This secretor, it will be having the antigen in the body fluid. So the antigen with the secretion or body fluid, it's said to be secretor. RH factor or RH antigen. RH factor, that is mentioning the RH blood group. RH blood group was discovered by Landsteiner and Alexander Weiner in 1940. They observed the RH factor first in rhesus monkey, that is Maca rhesus, and later they analyze in human being. The term RH factor, it is refers to the immunogenic D antigen of the RH blood group system. So where we can observe this RH antigen? It is observed on the surface of the RBC. The immunogenic D antigen in the individual are RHD positive, that is RH positive blood group, and those without the D antigen. If they are having the D antigen means it is RH positive. If they are not having the RH, that is D antigen means RH negative blood group. So this is the rhesus factor in the blood is inherited as a dominant factor. Naturally, the anti-D antibodies are absent in the plasma of any normal individual. So RH negative person does not contain the RH antigen. The RH antigen has no natural antibodies. The antibodies can be artificially produced. The RH person developed the RH antibody when he received the blood from the RH positive. The small amount of RH positive, it evokes the production of RH antibody in the RH negative person which will remain throughout their life. That is about the RH antigen. This RH positive person will be having D antigen. The RH negative person lack the D antigen. This is Fisher and Ray's hypothesis, where genetic control of RH factor is explained. RH factor involves 3 different pairs of alleles located on 3 different closely linked loci on the chromosomal pair. Here, the Fisher proposed that 3 genes are involved in the production of RH antigen, and these 3 genes are lying so close together on the chromosome, but they act as a one gene that is called as pseudoalleles. He proposed 3 dominant genes, capital C, capital D and capital E, and recessive allele, small c, small d and small e. So these are the 3 pairs of alleles. A RH negative person should have the genotype of recessive, small d, small d. RH positive person must have the dominant capital D. So if the capital D is found to be present in alleles, that represent the RH positive blood group. If it is a double recessive genotype small d will give rise to the RH negative phenotype. It's the Vienna hypothesis. He proposed the existence of 8 alleles, that is capital R1, capital R2, capital R0, capital RZ, small r1, small r11 and small ry. All these at a single RH locus. All genotype which are dominant is capital R, they will be producing RH positive phenotype, and double recessive genotype will give rise to the RH negative phenotype. So dominant character is RH positive and recessive character represent the RH negative phenotype. Erythroblastosis fetalis, or otherwise said to be incompatibility RH factor. Erythroblastosis is a hemolytic disease of newborn baby. It is characterized by jaundice and anemia due to the hemolysis. So, it is named because of the circulating erythroblast. Hemolysis is nothing but the death of RBC. Erythroblasts are normally found in the bone marrow. The presence of circulating erythroblasts account for the name of the disease. When an RH negative woman marries RH positive man, when the fetus which is developed in the womb is RH positive, it inherits the father's blood as RH positive. So, the RH positive baby developing in the uterus of RH mother develop erythroblastosis due to the damage of the blood vessel during the childbirth. The mother immune system recognizes the RH antigen and gets sensitized. The sensitized mother produces RH antibody, that is immunoglobulin G, IgG, gamma immunoglobulin. It is very small and it has the capacity to cross the placenta. It also enters the fetal circulation. When the mother gets sensitized, it produces anti-D antibodies at the time of delivery. Nothing will happen to the first childbirth. When she undergoes a subsequent pregnancy, carrying the same RH positive fetus, when the RH positive fetus exposed to the antibody produced by the mother against the RH antigen across the placenta into the fetal blood circulation. It causes the hemolysis of the fetal RBC. This results in hemolytic jaundice and anemia. That is said to be erythroblastosis fetalis or hemolytic disease of newborn, otherwise HDN. Now let us see how this jaundice is occurring. The destruction of the RBC leads to the hemolytic jaundice and anemia. So the destruction in the RBC is anemia. When it is entering into the liver for disintegration, which one? The dead RBCs. As more work is given to the liver is heavily damaged. When the liver gets damaged, it leads to the jaundice. This causes the death of the baby. Observe the picture showing the RH positive fetus inside the RH negative womb of mother. That antibody which is produced by the mother, it will be crossing the placenta. Observe the picture carefully. So which is the antibody crossing the placenta? Immunoglobulin G. That is immunoglobulin gamma cross the placenta. How to prevent the erythroblastosis? If the mother, that is RH negative mother, and the fetus is RH positive, anti-D antibodies should be administered to the mother at the 28th and 34th week of gestation, soon after the delivery. This will develop passive immunity and prevent the formation of anti-D antibodies in the mother blood by destroying the RH fetus before the mother's immune system is sensitized. This to be done for every pregnancy of the women.